So fast forward to 1952 and you get the Hershey and Chase Waring blender experiment. So this is further proof that DNA is the molecule of heredity and not proteins. So if you take these T2 bacteriophages, as Hershey and Chase did, which are composed of just two molecules, first the protein caspid, which is I've drawn in black over here, and then on the inside, it's their DNA. So the way that these bacteriophages work, they're just like viruses that infect bacteria. So if you have them infect E. coli, they would inject their DNA and then replicate their DNA within their host, and then after that, they would end up lysing the E. coli, and they would grow out as a bunch of more little T2 bacteriophages, and the cycle keeps going, infecting more and more bacteria. So what they did was take these T2 bacteriophages, and then they split them into two groups. One group was grown in this media, of course with E. coli, because you can't just grow bacteriophages. They won't replicate on their own, so they need to first infect E. coli so that they have a host to grow in, and then after that you grow them in a medium that has 35S labeled 35S, which is radioactive sulfur, and that is going to label your proteins so that when you shine UV light on them, they will fluoresce, and you'll be able to know, like, hey, that's where my proteins ended up. And then with the other group, what they did was grow it in a medium that contained 32P DNA. 32P, which is just the phosphorus, and as you know, that would label your DNA because you have phosphate in the backbone of DNA. So again, shine UV light, and then you'd be able to see the fluorescence and identify where your DNA is in this group. So what happened is they let each group infect their E. coli, and then after that they used their Waring blender to separate the ghosts, which is just the virus after it got rid of its DNA. I mean, they didn't know it was DNA, but after it transmitted whatever it is that it transmits into E. coli, and then after that, just that little empty bacteriophage is what we call the ghost, and then after that you have your infected cells, and they centrifuge that, and then the heavier infected cells go at the bottom, and then after that at the top you just have your ghosts. So in this case, they saw the fluorescence in the labeled proteins, or in the ghosts. So they knew that the protein was not going into the E. coli and infecting them or helping them replicate and pass on to future generations. So it was not the hereditary material. It just stayed in the hosts, which, aren't needed for the next generation. But then if you look at the sample that had fluorescently labeled DNA, you could see that the DNA, you see the fluorescence in the infected E. coli cells. And what's cool about that is that is going to be the DNA that's going to replicate within these hosts and then create your new bacteriophages. So this is further proof after Avery and his colleagues said, like, hey guys, it's not actually protein, it's DNA. Hershey and Chase further proved it, and then, the scientific community had to accept it at this point. Now we know that DNA is the molecule of inheritance.